Finally, in our sixth video of the unit, we begin talking about cellular respiration. Glycolysis is the first step of cell respiration, whether it's aerobic in the presence of oxygen or anaerobic in the absence of oxygen. This video is going to help accomplish these course goals. Pause the video and check them out. One of the tough things about studying cell respiration is that there are so many details to keep track of. In AP Biology, we'll learn to manage some of the details, but definitely not all of them. My goal for you is to have a solid foundation in cell respiration for the next time you have to study it, but have enough right now to do good in this class and on the AP test. The details you should concentrate on are listed on the slide. First, you should know the overall chemical equation of each step of cell respiration. Once you have this, you should be able to say where the reactants come from and where the products are going to. Finally, you should be able to tell the precise cellular location of each step of cell respiration. Let's start with an overview of the aerobic cellular respiration process. In aerobic cell respiration, there are three big steps. First is glycolysis, which happens in the cytoplasm. Next is the Krebs cycle, which happens in the mitochondria. And finally is the electron transport chain, which also happens inside of the mitochondria. Anaerobic cell respiration is a lot simpler. After glycolysis, there's only one other step in the cytoplasm, but we'll get to anaerobic respiration in the next video. Let's practice some of those big picture questions with overall aerobic cellular respiration to see what details are important. Remember, the three big steps of aerobic cell respiration are glycolysis, the Krebs cycle, and the electron transport chain. Since cellular respiration is breaking down glucose, cellular respiration overall is considered catabolic. The overall equation of cell respiration is shown here. Glucose plus 6 oxygens react together to give 6 carbon dioxides, 6 waters, and 36 ATPs. The amount of ATP can vary based on the organism and conditions of the cell, but we're just going to go with 36 to make things easiest. Now, these other questions that are on here, where do the reactants come from? Glucose comes from food, and oxygen comes from the environment. Can get up inside of the cells by breathing or by simple diffusion. Where are the products going? Well, for this overall equation for aerobic cellular respiration, carbon dioxide is a waste product that diffuses out of the cells by simple diffusion. Water can be kept around inside of the cell to do things, or if there's too much water, water can leave the cell by osmosis. And finally, the big goal of cell respiration are these ATP. Where are those products going? Those products are going to do work in the cell. Transport work, chemical work, or mechanical work. Now we're going to zoom in to the first step of cell respiration, glycolysis. If you dissect the word glycolysis, it literally means sugar splitting. In glycolysis, the sugar glucose, which is 6 carbons, is split to make 2 3-carbon pyruvates. One 6-carbon piece is split to make 2 3-carbon pieces. Glycolysis isn't just one reaction, but it's actually a process made up of 10 separate chemical reactions that we batch together to call glycolysis and make it easy to think about. Glycolysis occurs in the cytoplasm of cells and doesn't require oxygen. Any organism can do glycolysis. Finally, glycolysis is relatively inefficient, giving only 2 ATPs per glucose of the 36 ATPs overall in aerobic cellular respiration. Here's the overall equation for glycolysis. 1 glucose reacts to give 2 ATPs, 2 pyruvates, and 2 NADH. Where does that glucose reactant come from? Well, that comes from our food. Where do the products go? Let's take a closer look. The 2 ATPs are used to do cell processes, cell work. chemical work, mechanical work, and transport work. The two pyruvate products will move on to the next step. In aerobic cell respiration, the next step is the Krebs cycle. The two NADHs will be used to bring high-energy electrons to the electron transport chain in aerobic cell respiration. In anaerobic respiration, pyruvate and NADH have different pathways, which we'll get into later. The 10 reactions of glycolysis can be broken down into two groups of 5 reactions. The first phase of 5 reactions is sometimes called the investment phase, and the second 5 reactions are sometimes called the payoff phase. The investment reactions get their name because these first 5 reactions cost an investment of 2 ATP. Those 2 ATPs get glucose ready for the next reactions. Since there's a net input of energy, you can consider these endergonic. The payoff phase, however, generates 4 ATP. Think about 2 of those ATP going back to pay off this investment, and then you get a net of 2 ATP. The payoff phase is where you also get 2 molecules called NADH. NADH is an electron carrier that takes high energy electrons to the electron transport chain. I want to take a closer look at those first 5 reactions, the investment phase. You can see that the reactions on this diagram are labeled 1, 2, 3, 4, and 5, and the 6th one is in here just to show us a little bit of context. You can also see that each reaction has its own enzyme that's written in red writing. You can tell that these are enzymes because each has a name that ends in ACE. Reactions 1 and reactions 3 are the phosphorylation reactions that prime glucose with phosphates. The other 3 reactions, 2, 4, and 5, are involved in rearranging the structure of glucose and breaking glucose in half to make 2 3-carbon pieces by the end of this. Now remember, you don't have to know all of the enzyme names and the name of all of the glucose intermediates along the way. I wanted to show you these reactions so that you can see what happens. Also, think about the biochemistry of what's going on and how phosphates can be used to put bond strain on glucose. Remember, phosphates are negatively charged, and if you look at glucose down here, there are 2 phosphates on the glucose, one on this side and one on the other side. This means that the phosphates, both negatively charged, will want to repel and move away from one another. What ends up happening is glucose is kind of stuck in the middle, and there's a tug of war that creates bond strain, makes it easier to split glucose. Here's a cool diagram showing how the first phosphorylation of glucose works. The enzyme that catalyzes this reaction is called hexokinase. You can see that hexokinase has an active site with 2 pockets. One of the pockets fits glucose, and the other pocket fits ATP. Once in the active site, phosphate is transferred from ATP to glucose. Glucose is phosphorylated. At the end of this reaction, ADP and glucose 6-phosphates are the products that are released. This is the first investment of ATP. In a later reaction, glucose 6-phosphate is phosphorylated a second time on its opposite side. The second half of glycolysis is the payoff phase. In reaction 6, the first reaction of the payoff phase, 2 NADHs are made. Think about NADH as tickets or money in the bank that can be cashed in later at the electron transport chain. In reaction 7 and reaction 10, you can see that a total of 4 ATPs are made. 1, 2, 3, 4. 2 of these ATPs pay back the initial investment of ATP from the first 5 reactions, and then there are 2 bonus ATPs that can be used to do other work in the cell. I think it's important now to take a look at NAD and a related molecule called FAD. Here's a picture of NAD molecule. Both NAD and FAD are electron carriers in cell respiration. They take electrons from glucose and carry those electrons to the electron transport chain. NAD and FAD are the oxidized versions of the compounds. When those compounds become reduced, or when they gain electrons from glucose, that's when they become NADH and FADH2. Think about those electrons as being attached to the hydrogen, and that's why they get a hydrogen as well. NAD is actually more commonly called niacin, and you can find niacin on nutrition labels for a whole bunch of different things, including this 5-hour energy. I also think this is a great point to check out what's going on with redox reactions. In glycolysis, NAD is stealing high energy electrons from glucose. During this process, NAD gets turned into NADH and glucose gets turned into pyruvate. So what's being oxidized and what's being reduced in this redox reaction? Let's remember oil rig. Oxidation is loss. So what's being oxidized? What's losing electrons? Glucose. Reduction is gain. What gains those electrons from glucose? NAD gains the electrons. Remember, the oxidizing agent is the thing that's oxidizing glucose, which in this case is NAD. The reducing agent is the compound that's giving off electrons, that's reducing NAD, which in this case is glucose. Studying glycolysis is also a great time to make connections to evolution. Those 10 reactions in glycolysis are done by every living thing on Earth. Those enzymes are all conserved in all cells, in all living things. Humans, bacteria, yeast, panda bears, all of them have hexokinase and all of those other enzymes. They all use glucose to make ATP. And think about those conditions of glycolysis. Why does it make sense that glycolysis can happen in the cytoplasm? Well, because every organism has cytoplasm, but not every organism has mitochondria. If you look at life's timeline, mitochondria didn't come along until at least 2 billion years after the first cells. For bacteria to do glycolysis, glycolysis must happen in the cytoplasm. Why does it make sense that glycolysis happens in the absence of oxygen? Well, the atmosphere didn't have abundant oxygen until 800 million years after bacteria first came about. Here's what's the interesting part. The fact that all cells use glycolysis is further evidence of the fact that all organisms share the same common ancestor. Another glimpse into evolution can be seen if you consider the inefficiency of glycolysis. Glycolysis only harvests about 3.5% of energy that's contained in glucose. That's a huge 96% of the energy that's left. And this leaves space, or a niche, for organisms to evolve to do aerobic cell respiration. Still, aerobic cell respiration only harvests about 40 to 50% of the energy in glucose. All right, let's recap the basics of glycolysis. The overall reaction is here. One glucose reacts to form 2 pyruvates, 2 ATPs, and 2 NADHs. Where do the reactants come from? Glucose comes from the food that an organism eats. Where do the products go? Well, ATP is the easy one. ATP is going to be used to do cell work, but pyruvate and NADH have a little bit more complicated of a story. That ATP is going to be used to do cell processes. NADH can be used to go to the electron transport chain and carry high energy electrons there, maybe, if the conditions are right. And pyruvate, it's the same thing. It depends on what cell is there and what conditions are there. Let me explain more with NADH first. So you can see that NAD is needed to do step 6 of glycolysis. If all of the NAD is tied up as NADH, the NADH would need to drop an electron to become NAD again. NADH can lose electrons in 2 different ways. First, NADH can give its high energy electrons to the electron transport chain. Once NADH comes NAD again, this NAD can go back and help out with reaction 6. But organisms that can't do aerobic cell respiration and organisms that don't have an electron transport chain still need to regenerate NAD. In these cases, anaerobic respiration happens, or fermentation happens, where NADH gives electrons back to pyruvate in a process that regenerates NAD. We'll talk about these processes, fermentation processes, more in the next video. Likewise, pyruvate has different fates depending on the conditions of the cell. In organisms that can do aerobic cellular respiration and have oxygen present, pyruvate is moved on to the link reaction and the Krebs cycle and then the electron transport chain, which happens in the mitochondria of eukaryotes or in specialized areas of the cytoplasm and plasma membrane of prokaryotes. In organisms that can't do aerobic cellular respiration or when oxygen isn't abundant, then anaerobic respiration kicks in. Some organisms do alcohol fermentation as their anaerobic respiration, and others do lactic acid fermentation as their form of anaerobic respiration. Both types of anaerobic respiration happen in the cytoplasm. I don't want to lose the big point here, so let's take a second to recap. Depending on the conditions of the cell and type of organism, pyruvate is a branching point in glucose metabolism. Pyruvate can be used to do aerobic cellular respiration when oxygen is present, or pyruvate can branch over to anaerobic respiration when oxygen is absent. Some organisms can only do anaerobic cell respiration, and all of their pyruvate would go this way. Other organisms that do aerobic cell respiration will send as much as the pyruvate as they can over here, but if there's not enough oxygen, then the pyruvate will branch towards this way. So, organisms that can do aerobic cell respiration can actually do both and switch back and forth as needed. Here's another diagram showing the same concept. With oxygen, pyruvate is going to go down the pathway that leads through the link reaction and to the Krebs cycle. Without oxygen, pyruvate's going to undergo anaerobic cell respiration. Anaerobic cell respiration can be lactic acid fermentation, or anaerobic cell respiration can be alcohol fermentation. Let's recap the major points of this video. This video was on glycolysis, the first step of anaerobic and aerobic cell respiration. Glycolysis is actually made up of 10 different chemical reactions divided into a 5-reaction investment phase and a 5-reaction payoff phase. The payoff phase gives the high energy products of ATP and NADH. NADH and pyruvate are products of glycolysis that can go either to anaerobic or aerobic cellular respiration, depending on the organism and the conditions of the organism. Last, the fact that every organism does glycolysis using the same products, the same reactants, and the same enzymes is evidence that all organisms share a common ancestor.